Hello everyone. Today I am going to say you about the Mahlon Bush Hoagland was an American biochemist who discovered transfer RNA, that is tRNA, the translator of the genetic code. Mahlon Bush Hoagland was born in Boston, Massachusetts, US in 1921 to Hudson Hoagland and Anna Hoagland. Hudson was an American physiologist who was known for co-founding the Worcester Foundation for Experimental Biology with Gregory Panics. He graduated from the Hill School in 1940 and attended Williams College, and in 1948 received his MD from Harvard Medical School with intentions of becoming a pediatric surgeon. After a bout with tuberculosis, Hoagland was forced to change career directions and became involved with research. After graduating from Harvard Medical School, he lived with designer Lois Cannon and her family. Cannon was part of the Follico designers, and Hoagland worked with them in Gloucester for several years while he commuted to Boston for work. Hoagland took a research position at Massachusetts General Hospital in the lab of Paul Zemsnik, where he researched and detailed the role of transfer RNA in forming proteins. He was working with Zamsonic and Elizabeth Keller when he discovered the initial steps of protein synthesis. From 1953 to 1967, Hoagland served as an associate professor of microbiology at Harvard Medical School. In the year 1967, upon leaving Harvard, he was appointed professor in the biochemistry department at the Dartmouth Medical School. In 1970, Hoagland became scientific director of the Worcester Foundation for Experimental Biology, retiring in the year 1985 after 15 years in the directorship. By the time Hoagland had joined Huntington Laboratories, his colleagues there were well known for their work in the protein synthesis. In the early 1950s, Hoagland and his associates were able to show that polypeptide synthesis occurs on ribosomes. They did this by giving rat injections of radioactive amino acids, waiting for a defined period of time, extracting the liver, and examining subcellular fractions for radioactivity. He found that after longer periods of time, radioactively labeled proteins were present in all subcellular fractions. However, if they allowed less time to pass, radioactivity was found in only certain particles, which they deemed the sites of protein synthesis. These particles were named ribosomes. During their experiments, the rat liver cells, Hoagland and Zamsonic noticed that in the presence of ATP, amino acids associates with heat-soluble RNA, which was later named transfer RNA, that is tRNA. This amino acid and tRNA complex, later called aminosyl tRNA. Hoagland's major contribution to the laboratory was in his work with amino acid activating enzymes. He discovered that certain enzymes were required to activate amino acids so they could associate with tRNA molecules and eventually be incorporated into new protein molecules. These enzymes were named aminosyl tRNA synthetases. Incidentally, this lab's discovery of tRNA supported the theory of complementary proposed by Watson and Crick. He was awarded the Franklin Medal in 1976. Mahlon Hoagland's other work involved the carcinogenic effects of beryllium, biosynthesis of coenzyme A, as well as liver regeneration and control. Thank you everyone.